everyone, welcome to the ATAR Notes Bio 3-4 lecture for July. We're going to be talking about all things sort of related to Unit 4, particularly the first area of study, so a lot about immunity today. My name's Loz, I'll be running through the lecture with you. Before we do get into it though, I would like to firstly acknowledge the traditional owners of the land from which I'm presenting from today, so the Wurundjeri and the Bunurong people of the Kulin Nations, and I'd like to pay my respects to their elders, past, present, and emerging. So a few of you may be familiar with some of the lectures. You may have attended some earlier this week or, in previous years or earlier this year as well. Essentially, ATAR Notes runs these lectures for, VCE and a couple of other states and across Australia. We do have a lot of other resources that essentially are made for high school students by past students. So that includes the lectures, it includes a lot of other things as well. So some of the stuff you see here, you may be pretty familiar with our study notes, also the discussion forums. Especially if you guys are in year 12, you may be using things like ATAR calculators. Obviously a lot of, revision stuff coming up into this next term. But yeah, if you do find yourself, being interested, feel free to head to the ATAR Notes website just to have a look at anything that might be useful to you. Bio is a really, really popular subject, and a lot of people take it, obviously. So there's a lot of resources available. There's a lot of input from past students and lots of resources kind of available, especially due to the new study design change. You guys are the second cohort to use the study design. So I think there's a lot of useful information on there, maybe from past students who have done it last year as well. If you do find yourself being more interested in some of our other resources, we do have a couple more. So TuteSmart is the tutoring company run by ATAR Notes. So I tutor bio there, for example. And there's a lot of other subjects available that you guys may be taking. Again, if you're in year 11 or year 12, I don't know, maybe year 10 even, there, maybe something there available for you. Otherwise, if you are interested in the study guides that I was mentioning, you can get hard copies. You can also get copies of, course notes, topic tests, some NEEP past exams as well, all available on Ed Unlimited. It's sort of like a little Netflix, but for our ATAR Notes study resources. So you can access it on your laptop, on your iPad, all that sort of stuff. And then, you just have access to every ATAR Notes publication. So if you do think that is something that might interest you, feel free to head to ATAR Notes and you can register an account and have a look at all of these things as well. Okay, so for our lecture today, we're essentially going through three major sort of blocks. Again, it's all to do with this first area of study of Unit 4. It is quite a bulky area of study. I know, some of your schools may have started with immunity already. Some of you may be going into it, in this upcoming term. It is a big area of study. There's a lot of stuff and it can get complex at times. I feel like sometimes it's regarded as, the most tricky subject. Again, it depends on what people say, but I feel like sometimes it's a little bit notorious. I don't think it needs to be. I think it is relatively complex, but once you break it down, it can be easier to understand. I think for me, it was probably the area of study that I enjoyed the most, I would say. Maybe that or like area study 1 of Unit 3. But I really liked it. And I think it was definitely hard to understand, particularly the adaptive immunity stuff. But once you sort of break it down and hopefully once we explain some of the common pitfalls today and some of the, common misconceptions that students might have, hopefully it'll be a little bit easier to grasp. Already in these past 6 months, you would have probably picked up on some study strategies that help you. For this area of study, I really highly suggest a lot of videos or sort of visual stuff. Again, I know people have different learning types, but just because of the nature of immunity and the fact that it's on such a molecular level and there's lots of, processes and sort of steps involved, sometimes watching a visual kind of explanation of that can be helpful. I know I found it really helpful. So like a lot of videos, even drawing diagrams, you can use others, but drawing your own diagrams can really help explain processes as well, going along with that, a lot of flow charts, that sort of thing. I feel like in bio in general, it's pretty helpful, but particularly for this area of study, or at least this first block in particular, kind of explaining your first, second, and third lines of defense, it can be really, really helpful. In terms of the experimental design, again, schools may do experimental design in terms of your actual SAC at different times. However, it's so important to keep on revising this for your exam. In saying that, while you're at this point of the year, you've just finished Unit 3, it's quite important to be revising all of this information as you go into Unit 4, because the last thing you want is to sort of get to, September, October, and you haven't looked at anything to do with transcription, translation, cellular respiration in a couple of months. Because then you're going to have to sort of waste time going back and almost like learning it again when you could have been just revising it sort of infrequently throughout this term, just to sort of maintain that knowledge. I feel like it'll help you in the long run. Yeah, those are kind of the main things. We'll talk about some exam tips at the end. If you have any questions, please pop it in the live chat. I'll help you, answer them and all that sort of stuff. Again, there is quite a bit of content, but I think we'll be able to go through it in hopefully a good amount of depth today. But if there's anything that you don't get, obviously feel free to ask a question. But also these slides will be available to you, and you can also email me as well. It's just lourdes@tutesmart.com is my email. So if after this, I'll remind you guys at the end, if there's anything that you feel like you missed or you'd rather email me about, feel more than free to send me anything. But yeah, those are probably the main things, but we'll obviously explain more as we go through it. Okay, so the first part of the area of study is looking at kind of immunity as a whole. So we have our little study design dot points that will appear in the corner, so just refer to them. As I mentioned this, the study design is really, really, really important. I've already discussed how you guys are ultimately the second cohort to use this new study design. I guess it is, I feel like being at the end of study design is definitely a bit better than being at the start, just because you've got past practice exams and things like that. But there was, I would say, more stuff taken out of this study design than was put in. So I think that's a good thing that you guys can sort of work with and use to your advantage. In terms of immunity, not a lot of stuff was changed. I think most of the stuff that's in the study design at the moment was in the study design previously, but again, just some things have been cut out a little bit. But yeah, use the study design to your advantage. I would try and not memorize it word for word, but definitely have an idea of all the dot points that are in each area of study, because this can be really helpful when you get to your short answer questions, especially when you don't know what the question is sort of asking of you. Sometimes you just get to a question and you read it and you just don't know, honestly. And you don't know, what it's trying to ask you. You don't know what it could be referring to. And at that point, speaking from kind of my experience, when I get to a question like that, I always just try to think, I almost like run through the study design in my head and I think, what dot point could this possibly be referring to? What dot point are these, VCAR examiners, what are they trying to kind of nudge me towards, if that sort of makes sense? So I feel like having a good grasp of the study design can be really helpful for that. But I'll talk about that, I guess, a little bit later on. Okay, to start with our actual stuff, we're discussing pathogens. So it's important to understand the distinction between different types, how antigens kind of appear on pathogens and that sort of thing. Definitions. So this is sort of the definition in terms of biological agents that can cause disease, basically. So they impair the normal functioning of the host organism. Obviously, they replicate, and that's how they can kind of spread their disease and how a disease progresses, essentially. And this idea of separating our pathogens between cellular and non-cellular, also very important, because VCAR will, include questions about this. In terms of definitions, sorry, I feel like I'm putting a lot of tips in before I'm getting to the actual content, but definitions are super important in bio, more so than other subjects, I would say. I remember my teacher always going on about keeping a glossary. And I think it is something that's really important because a lot of the questions in bio are definition based. So particularly, it can happen in multiple choice as well, but particularly a lot of short answer, they can use basically like a lot of marks are associated with using a definition. And I feel like I didn't really like making a glossary, particularly early on. So I used to kind of like ignore my teacher and be like, oh, whatever, like a glossary, like, because I hadn't used a glossary in the past. And I feel like I was very sort of stuck in my ways when I was entering year 10. I realized I didn't really say much about how experienced bio, but I did bio when I was in year 12. So I did 1 and 2 in year 11. I did 3, 4 in year 12. I got a 45 in bio, and I did enjoy it, I would say. I think I enjoyed 3, 4, I would say a lot more than 1, 2. Um, yeah, and I think I was able to do well in it because I sort of was able to figure out, and I think this is a really important point, how to answer bio questions. I always say to my bio students, it's a completely different thing knowing the bio content and being able to score well, or not even score well, but being able to answer bio questions properly and getting the full mark for the question. Um, Again, talking about how I was always stuck in my ways, I never really did, practice questions and glossaries and that sort of thing. I was very much, do your chapter summaries and then, you know, write your notes and then you answer the question, you get it right. But in bio, I found that wasn't exactly true. I would do my chapter summaries, but then, because I think I would get a pretty good grasp of the content, but then initially I basically wasn't answering the questions properly. I think bio is very specific in the way that examiners want you to answer questions in a particular way, and there's definitely a very particular marking scheme. Again, I would say more so I found it to be different to some of the other sciences where maybe that technique, my old technique that I used to use would work fine. But I think for that reason, having a glossary, doing your practice questions, it's all really important because you can know the content like the back of your hand, but if you don't understand what the question is asking of you and you don't know how to phrase all the knowledge in your head properly, it's not going to work out and you're not going to get full marks. So that is something really, really important to be aware of. Okay, I think I've got enough tips to share now. We'll talk about other things later. But in terms of the actual pathogens that you have to know, bacteria are a really important one. They're probably the most common one that pops up on your exams. So unicellular prokaryotes, so they are cellular, but it's just one cell basically compared to your sort of eukaryotes and your more complex ones. So prokaryotes, basically they're just very, very simple organisms reproducing independently. So via binary fission, that is a big problem for them. I mean, I guess for us because of them, because of that idea that they can reproduce really, really quickly. So obviously we think about pathogens and bacteria in this context of spreading disease, but bacteria are really helpful. We obviously, we'll talk about our normal flora and stuff and how that can form a bit of a defense system. But yeah, you've got bacteria all over you, your skin, your gut, everywhere, mostly everywhere. But in that certain case, they're not pathogenic because they're not causing disease. In fact, they're doing the opposite. They're being helpful. So when we talk about pathogens, it's in that context of causing disease. Sometimes you might call them opportunistic pathogens. So that's when bacteria that perhaps exist on your body that don't usually cause disease, suddenly they get the opportunity to, and then they do so. So that's just a bit of an example there. In terms of the cell wall, this idea of peptidoglycan, it's not something vital to know, but it's something that is important for certain bacteria, and it can be a good target for a lot of antibiotics. And we classify them based on their shape. So you can see you've got cocci, you've got rods, spirochetes, all these different types. It's not vital for you to know, what's what, and you don't need to know specific examples of bacteria, but just understand that they come in different shapes. Typically, the ones used on a lot of Beaker diagrams and stuff like that are your rods. But yeah, there are different types, so just be aware of that. Be aware of some of the common sort of like characteristics. So you've got you can have like flagella, so this thing that almost like a little bit of a tail. You can have your pili, which sort of stick out. Again, you've got your cell wall, you've got like a capsule sometimes. And so all of these things can be sources of antigens. So we'll get into antigens in a second. But basically just these things that can almost be like little tags on bacteria that our immune cells can recognize. So it's important to be able to recognize, these things like flagella and pili, so you can see it on a diagram and know that you're looking at a bacterium, but also so that you understand that, the flagella may be part of an antigen that an antibody is specific for example. But yeah, just get very comfortable with knowing key things about bacteria and knowing what they look like on a diagram as well. Okay, so ways that they sort of work: producing toxins, invading tissues, consuming nutrients. So, you've got your exotoxins, your endotoxins. Not a vital distinction to be aware of, but just the idea of exotoxins being secreted while the bacteria are alive. And obviously, the toxins can be harmful to our cells, or, the host organism cells. And then your endotoxins are basically when the bacteria bursts, when it dies, the toxins that are inside, as in endo, they leak out, and then they can obviously cause a lot of damage as well. Um, so invading tissues. So we kind of think of bacteria as extracellular pathogens. So they exist sort of outside the cell, and then they can come in and invade tissues. They can release these enzymes, release these other toxins that ultimately break down that tissue and break down the cells in that tissue, and that's obviously going to lead to, consequences, symptoms, and ultimately sometimes death of those cells and those tissues. Consuming nutrients is a big thing because basically they just outcompete everything else. So you've got your bacteria and they're consuming nutrients that your cells need. Your cells are losing, they're going to die ultimately. So that's often why in bacterial infections you can get changes in the level of glucose, because of this idea that bacteria are consuming the resources that your cells need. And that's kind of pretty general, inhibiting normal cell functioning as well. Okay, hopefully that all makes sense. Viruses are your kind of second, main pathogen that VCA often use. So definition here: obligate intracellular pathogens. So obligate is the way that because viruses are non-cellular, very important to understand that as well. Non-cellular, please remember that. They can't reproduce by themselves, which is why they are an obligate intracellular pathogen. They rely on being inside a cell and having a host and having something that transcribes and translates in order to replicate. So that's why they're an obligate. They can't sometimes invade a host cell and then sometimes they can just reproduce by themselves. They have to basically hijack the machinery or the replication system in order to replicate and then spread those virus particles to other cells. Okay, so then as we've mentioned, so they will basically infect a cell, transcribe, translate the genetic information that sits inside of their little capsid, and then produce more virions or viral particles. Those will then leave the cell, infect another cell, and that's how the infection spreads. And at the same time, they can go around killing those cells as well. So there are heaps of different viruses. You can be based on their genetic information. So they may have DNA, they may have RNA. Bacteriophages they look like little spiders. You've probably seen it when you looked at CRISPR, but they are very specific to bacteria. They aren't really, they don't really infect human cells, so we don't see them as often in our examples or in VCAR questions. But it might be good to recognize, especially in that context of CRISPR. And then, there's other different types of viruses. Again, no virus that you need to know specifically. Some viruses will come up more than others. You probably get a couple questions on COVID, HIV, those sorts of things. But there will never be a really specific question about a certain virus because it's not on study design. You know what I mean? You have to know viruses generally, but they'll never ask what is the exact specific mechanism of an RNA virus or HIV, or describe all of the different antigens of COVID and stuff like that. This is another common thing, a little tip with bio as well. You'll often see things that are unfamiliar. Don't let it psych you out, so you kind of get lost and think, oh my gosh, no, I haven't revised this. I didn't look at this specific example of HIV, or I didn't look at this specific example of COVID. Because VCAR often will try to distract you, especially in multiple choice questions. They'll give you this whole chunk of information. They'll talk, all about COVID and this and that and blah, 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 all this random information. And you read it and you'll go, oh my gosh, no, I haven't looked at this before. I've never heard of this before. I'm not going to be able to answer the question. And then basically the hidden question will be are viruses, cellular or non-cellular? But they'll sort of hide it amongst all that information. So then students who aren't aware of what they need to know and what they don't need to know might get frazzled. So again, why the study design is important, because in questions like that, you don't need to worry and go, oh my gosh, I've missed something. Because you'll be able to read the question and understand that, they're just providing some extra context and almost trying to distract you a little bit. Okay, so prions are another type of pathogen that we can see down the bottom there. They are also non-cellular. They are ultimately misformed proteins. So prions basically they're like yeah, misformed proteins. You see them often. The disease may spread from animals. So if you have these really harmful misformed proteins, and let's say you get infected with one, so say you may eat meat that's infected with a prion, right? So you'll eat this and you'll obviously eat the protein, and because it's a misformed protein, it will come into contact with the sort of normal version of the protein in your body, and then it will like convert it. It will change that normal protein to this misfolded protein, and it's really harmful. It's kind of affects the nervous system, so you get some of the neurological disorders. So if you had like mad cow's disease and stuff like that, um, that's the kind of sort of things you're thinking about with prions. So they are non-cellular. It's important to remember that viruses are non-cellular and prions are non-cellular. But your viruses are, you know, your little hijacking genetic information sort of thing. And your prions are just misfolded proteins, basically. And so prions are thinking more neurological disorders. Prions, I would say they honestly don't come up very often in exams, whereas viruses, you'll be talking about them every second immunity question. Okay, so here's a nice diagram that sort of summarizes this. You don't need to know the cycle of a virus, but it just may help you visualize it. So you've got your virion, which is just a virus particle that enters the cell. You can see its genetic information held sort of within that the center of that particle, and you can see it's like little proteins, spike proteins on the outside. This is a very typical diagram, like as in this of what a virus will look like. You can expect it to look like that in your VCAR exams. But it basically, yeah, will insert its genetic information into the nucleus. We've got transcription going, and then we'll have translation going. Those proteins that genetic information codes for, which is obviously just to create more virus particles. As you can see here, we've got our spike proteins and stuff like that. Those will be translated, and then you get this new virus particle forming. It ultimately exits the cell, and then this will go on and infect another cell. So that's the idea of how viruses work. Super important thing, just remember that it's non-cellular. Okay, so some of our eukaryotic pathogens, so these are cells that are a little bit more similar to our cells, so a little bit more complex. So we've got our fungi, so they, you think of like your fungal infections ultimately, so like your tinea, your athlete's foot, that sort of thing. Their cell wall is made of chitin, so you can think again, just like peptidoglycan with your bacteria. The chitin is sort of another target that you could use. But yeah, those are your sort of fungal infections. And then your protozoa. So these are your eukaryotic pathogens, but they're single-celled. So I feel like often we tend to think like eukaryotes are multicellular and prokaryotes are single-cellular. Remember that that's not always the case, such as these protozoa that are eukaryotes, but they're a little bit more simpler. They're still unicellular. So these can sort of be quite parasitic. So like Plasmodium is the cause of malaria. So often we think mosquitoes are the cause of malaria. They're the vector. So just like, in Unit 3 when you discuss plasmids and all that stuff and how your plasmid is the vector for sort of carrying your target gene into your bacterium, your mosquito is basically the vector of carrying this protozoa, this Plasmodium, into the human, let's say. So yeah, that's the pathogen we're thinking about. And then worms, so what we've got depicted here, your tapeworms, your liver flukes, that's pretty self-explanatory in terms of what they are. They just look like real worms as well. But they can be obviously a lot bigger, and yet that sort of parasitic nature, and where they can just take up the nutrients and absorb all of that at the expense of the host. Okay, so those are the pathogens you need to know. Really important. Again, bacteria and viruses, the main ones you sort of think of. Okay, so in terms of our actual sort of immunology stuff here, so an antigen, very important, again, definition to know. So it is a molecule that is capable of inducing an immune response. So we've got our self and our non-self antigens. So on our pathogens, they will have their own little antigens. If we're thinking of a bacteria, maybe just a little protein on the capsule, or if we're thinking about the virus, like a spike protein, that is the antigen. It's just like a little, I like to think of antigens as name tags. It's just like a little name tag basically identifying what that pathogen is. And if you think of a self antigen, so like your MHC markers, these are again little name tags that say, hey, like I belong to the body, basically. So your MHC markers are your MHC1 in particular are basically your self antigens. So they are found on all nucleated cells. So be aware that they aren't on, certain cells like red blood cells and things like that, but they have their own little, like they've got their ABO system. So there's still ways of identifying cells there. But your MHC1 are essentially, your little name tags. And the way that they work is, and I think this is important to know. I remember I didn't know this, this sort of second dot point here. I didn't know it until we were kind of further into the area of study. And I think to me, it helps everything click a little bit more, especially when you think about the difference between cytotoxic T cells and natural killer cells. Knowing what MHC1 actually does, as opposed to just being a self antigen, for me was really helpful. So just bear that in mind as we talk through them. So with the MHC1, I like to think of MHC1 as basically little windows. I think I might elaborate on this when we get to natural killer cells and stuff like that. But they basically show immune cells. So I like to think of so cells as houses, your MHC1 as windows, and your immune cells as police. So your immune cells go around and they just basically double check everyone's antigens and make sure that there's no foreign antigens hanging around that they need to be a little bit wary of. So the way that they do that is through these MHC1 markers. So that's why I like to think of them as little windows, because basically the police, so the immune cells, they run around and they look in everybody's window. So they look at these MHC1, and MHC1 shows what proteins are being made in the cell. So they present peptides derived from cytosolic proteins. So your cytosol, proteins that are hanging around in your cytosol, they take like little bits of that and they put them on their MHC1, and then that shows the immune cells, this is what I'm up to inside of my cell. So that's why I think of them as a little window. So then if something is going a bit dodgy in there, in your MHC1, then the police can realize and then they can sort it out. Yes, that's what I like to think of it as. So obviously if you're making the proteins that you're meant to be making, that's how MHC1 works, is basically a self-antigen, a self-marker. It tells the immune cells, I am a cell from this body. I'm meant to be here. I'm not doing anything wrong. Your class 2 are a little bit different. They are associated with antigen-presenting cells. So not every single cell has MHC2. Only specific immune cells have it. And these will be used, like I was going to say window again, but not really window. These will be used basically just to show when you've got a foreign pathogen hanging around the body. Okay, so we'll show a little picture. So here is this idea of self-antigens and non-self antigens. So your T cell, which we'll get into in a little second, is a really important immune cell. So they go around and they, can check what's going on. And it's this idea that they have different receptors. If there's a theme for immunity, it's receptors, this idea of things being complementary, matching up, detection, all that sort of thing. A very, very important sort of theme of immunity. So your MHC1 markers, as we mentioned, are your self-antigens. And then your, stuff that might be on a pathogen is your non-self antigen. So T cells are able to basically detect what belongs in the body and what doesn't. So hopefully that makes sense. Okay, so to get us sort of thinking, so when a virus attacks or infects a human cell, what occurs within the cell? So again, think about your viral particles. Think about what our definition was. So an obligate intracellular pathogen. I was thinking parasite. Yeah, obligate intracellular pathogen. So we're going in a cell and we're basically hijacking that machinery. So we are undergoing transcription, we're undergoing translation, and that cell, so our human cell, will begin to become basically a virus making factory. So MHC1 markers, what they present on the outside of cells. So hopefully you guys are all thinking of the little analogy of a window. Okay, I don't know if it's helpful. It was helpful for me. If you think it's stupid, don't use it. Think of something else. But yeah, I just like to think of them as a little window. So they present your little proteins that your cell is making. Again, just a way of showing the immune cells that you're sort of doing the right thing. If you think about it, with a virus, if you're infected with a virus, what you would be making, if you're making viral proteins, you're going to be essentially displaying that on your MHC1. So that's this idea of how the immune system detects cells infected by viruses. Your MHC1, so your windows, they'll be displaying your viral particles. So you've got, I don't know, a home invasion, right? The virus is a home invader. I guess they come in, they start invading, and your windows are still open, and the police come around, your immune cells come around, and they can see through the window that there is someone invading your home, and then they sort it out from there. So that's the idea of how immune cells detect when cells are infected by viruses, via their MHC1 markers. Okay, so looking at our first line of defense, so basically first line of defense, I think it should be used kind of colloquially. According to the study design, it's better to say, your innate and your adaptive immune response as opposed to first, second, third. I think that's something my teacher used to tell me as well. So just be aware of that. Sometimes it's easier to sort of comprehend your first, your second, and your third. But technically, like when you're writing, in short answers, I would never really write first, second, or third. I'm sure depends how lenient your examiner is and all that sort of stuff, but it's always best to use terminology that's listed on the study design. Okay, so you've got your non-specific or your innate immunity and your adaptive or your specific immunity. So your non-specific immunity, and it's important to know differences between these. Your non-specific immunity is basically your one size fits all, works really quickly, sort of like instant, like putting a band-aid, ultimately. So whatever the pathogen is, if it's a virus, if it's a bacteria, if it's a prion, if it's a whatever, the innate immune system is going to act in the same way. Again, one size fits all. There is no memory of prior pathogens. So this idea of memory will make more sense when we talk about adaptive immunity, but it's this idea that it's one size fits all, and the cells that are involved with this, you could be infected with the same bacteria over and over again, but they won't recognize it, is the idea, whereas in adaptive, you do recognize it. And then the level of response being the same for each pathogen of the same organism. Again, that idea, if you're infected by the same bacteria over and over again, when innate immunity kicks in, it'll just kick in. It's one size fits all, exactly the same, copy and paste. Whereas with adaptive immunity, because you have that memory of past infections, if the same organism comes along, that memory kicks in really well and you get a bigger response, and then you get a bigger response, and you get a bigger response every time something, the same organism tries to infect, ultimately. Okay, so in terms of the first line of defense, so this is preventing pathogens from entering the individual, really. A big one is intact skin. You can see it's underlined intact. If you just say skin, you will not get the mark because the skin could be open, the skin could be damaged. Intact skin, it has to be closed. So that's obviously a pretty clear physical barrier. And that's why when it's open, that, skin is sort of useless if there's a big gaping hole in it, because then the bacteria can just jump right in. So mucus and your cilia in the airways. It's also important to know what is a sort of physical and what's a chemical, I believe. But I think it's in the other dot point. But your physical, your chemical, and your sort of microbiota, it's important distinctions to make. So your intact skin would be more physical. Your mucus and your cilia can ultimately be both a little bit. Basically because the mucus, I prefer to think about it in a physical sort of sense because I think it's easier to explain in a short answer. Your mucus can ultimately trap those pathogens, and then the cilia just pushes it so it gets sort of chucked, back out or like swallowed and stuff like that. But there are chemicals within the mucus as well that can help to sort of degrade that pathogen. So if you're going to describe it in, a chemical aspect, just make sure that you're being specific, and be mindful that VCAR may ask you, what are some examples of physical barriers? What are some examples of chemical barriers? And you need to know different ones. So a low pH in the stomach, so this is chemical. Stomach acid is very acidic, so it can kill certain pathogens, but of course there are some bacteria that hang around in the stomach as well. So lysozyme, so these enzymes in your tears and in your saliva, they can help to degrade certain pathogens as well. So your good bacteria, this should be referred to as your microbiome, your microbiota. So they're found in other places on your body. It says a few exceptions, those are just sterile areas, but it's this idea that this microbiome helps because they compete with your bad bacteria. It's like a little bit of a good versus bad. So when let's say you have a poor microbiome, so I was talking about opportunistic pathogens before. If you take some antibiotics, let's say, they may be pretty general, and what they might do is they may end up killing some of the good flora, or like the good bacteria in your body. And what that means is there's sort of less competition. And so sometimes you can get other opportunistic infections. Often like thrush can be an example. So sometimes you have a fungus that sort of sits around your mouth, for example. And sometimes if you take antibiotics, or even if you take like Ventolin, like if you take steroids, then you basically, kill some of the bacteria around, and then those pathogens that are left, they can become opportunistic and kind of grow. So that's why it's really important to keep a nice balance of the bacteria in your microbiome for that reason, because the competition is really helpful in making sure that they don't get too crazy, but then also that, your actual foreign pathogens don't invade as well. Okay, so in terms of your second line, so let's think, we've gotten, something's gone wrong with our first line. Maybe we have a big cut. Now we've got pathogens that are entering our internal environment. So this is when the second line kicks in. This is your innate immunity. So these are your sort of general leukocytes. Again, this is your sort of one size fits all. So this is a nice little map of your leukocytes. The ones that are blocked out are part of the third line, so your adaptive immunity. So we'll look at those a little bit later. But these are the really important cells for you to be aware of. Be aware, because I feel like sometimes I say it and it might not be captured as well. So this is a good diagram. But you'll often see this thing like a monocyte, and they'll often link it to macrophages. So monocytes are just like an earlier version of macrophages. I think it can be confusing. Sometimes students think it's two different things, but you've got your macrophage, which is a really big like main character in the innate immune system. And then your monocyte is just like the baby version of that, basically. Okay, so this is a really, really important table, or just an important thing to be aware of. Flashcards can be good for this, diagrams can be good for this. Whatever it is, just know the main features of these key innate immune cells. So your macrophages, basically we're thinking about them being really good at phagocytosis. We'll discuss phagocytosis in a second, but it's basically just engulfing pathogens. Engulfing pathogens. As in like phagocytosis, like, like exocytosis and endocytosis. Phagocytosis, like they're just like eating the pathogen basically, and they degrade it and chop it up. Your dendritic cells, I should mention as well, they are an antigen presenting cell, so an APC, so that's why they've got MHC class 2. Your dendritic cells are APCs as well, and they're like the best of the best. So your macrophages are very well known for phagocytosis. Your dendritic cells are very well known for antigen presenting. So that's something to be aware of. Neutrophils, these are not antigen presenters, so they do not have MHC class 2. They're the most common type of leukocyte that you see sort of in infections. So there's really, really high numbers of neutrophils. And then they ultimately do this like little kamikaze thing. So they undergo apoptosis after phagocytosis. So with macrophages, when they phagocytose something, they eat it up, cool, they move to the next one, eat it up, move to the next one, eat it up, move to the next one. Neutrophils, kind of like when a bee stings someone and then they die, that's sort of the idea with neutrophils. They will engulf a pathogen, and the way they kill it is that they release all of these like really harmful enzymes and toxins or whatever. So it kills the pathogen, but it ends up killing them in the process. So that's kind of a little characteristic about them. Mast cells, really key. You always associate them with histamine, and they're very involved in your sort of allergic and inflammatory responses. Something to be aware of is that they reside in the connective tissue. So they're not kind of floating throughout the bloodstream. They just sort of sit and release the histamine. Natural killer cells also quite important. These work similarly, not the same, but similarly to cytotoxic T cells. So it's important not to get them confused. And they release these two things you have to know, perforins and granzymes, which you can see here. And those basically induce death in certain cells. So they will detect changes in your MHC class 1. So the way that they do this, remember that innate cells are a one-size-fits-all. So what certain pathogens might do, so some viruses and especially some cancerous cells as well, they know that, the police are coming around looking through windows. So what they do is they shut the window. So the MHC class 1, they down-regulate it. So they just don't have an MHC class 1. This is what the natural killer cell will pick up on. So the natural killer cell, again, one size fits all. It's not checking, is this chickenpox? Is this COVID? Is this salmonella? It doesn't care what it is. It just goes around from house to house, and anybody that has their window closed, as in any cell that doesn't have an MHC class 1, they will release these perforins and granzymes, and that will induce apoptosis, so cell death, and that will kill the cell ultimately. The cytotoxic T cell is a little bit different because it's part of the adaptive, so the specific immune system. So we'll talk about that when we get there. But just remember that natural killer cell, just like all of these cells, they don't care what they're killing. They just know that it's a pathogen. Whereas the adaptive system is a little bit different and more specific. Okay, and then lastly, your eosinophils. These are kind of like the random, like nobody really cares about it. But important in your parasitic infections, and they can undergo phagocytosis as well. Yes. Okay. So we have a lot of like little sidekicks that kind of support these immune cells. So your complement proteins, these are really important. So they basically just help these immune cells. So they can enhance phagocytosis. They can bring immune cells closer to the site of infection. They can actually lyse some bacteria themselves. So they form this thing called a membrane attack complex, and they just kind of like sit on the like plasma membrane or the membrane of the pathogen, and they form a little hole, and then it causes all of the contents to sort of leak out of the pathogen, and then they burst and they die. And then they can help sort of work with antibodies as well, which we'll discuss soon. So cytokines are also quite important. They're signaling molecules, and they're basically just methods of communication. So if a cell wants to talk to another cell, they'll release these cytokines. So cytokines are cytokines, really important. Interferons are also very important. They're a sort of subset of these cytokines, and they're released by cells that are infected with viruses. And what they do is they basically warn the other cells nearby, and they tell them, I'm infected by a virus, watch out. What they also do is they will release, if I'm a cell that's infected by a virus, I'll release this interferon and I'll send it to my friends, right? Telling them be careful. But what I'll also do is I'll send it back to myself. So I'll release it and it'll bind to a receptor on my membrane, and it'll tell me like, slow down with your protein synthesis, like you're infected by a virus, relax a little bit. So that's why interferons are really quite important in viral infections. Okay, this is also extremely important, your inflammatory response. This ties in as well with allergies, which we'll get to. So your inflammatory response is part of your innate immune system. So again, it's this sort of one size fits all idea. So we've got damage occurring to the site. So this is our intact skin. We've now got a hole. That first line is breached. So these damaged cells release chemical signals. So the damaged cells around here will detect that they've been, like the cells in the tissue here will detect that they've been damaged, and that will sort of call your innate immune system into play. Remembering that your innate immune system is very quick. So your macrophages are, ready. They're going, okay, I'm coming to the site of infection, time to help out. Your mast cells, which are these ones here, remember that they sit in the tissue. Unlike these other cells, they're not running through the bloodstream. These ones don't move. They sit in the tissue, and they'll kind of recognize like, oh, something's going on here, and they will release these molecules here, the histamine, and the histamine really gets everything going in this inflammatory response. So we have our main sort of symptoms, so redness, heat, swelling, pain, and loss of function. It's like a thing, they're like your 5 like cornerstones, like major things of your inflammation. And they all kind of come as a result of this sort of histamine and this idea of getting your immune cells into this location. So the redness is because you dilate your blood vessels. So your blood vessels become leakier, they become more permeable, they become wider, so vasodilation, and that just increases the blood flow and allows more immune cells to get to the site quicker. The heat is because of that as well, because your blood is warm. The swelling is because your blood vessels become, as I mentioned, like leakier and more permeable, and this allows immune cells to squeeze through the gaps a little bit more easily. So that idea of them becoming more permeable, means that your immune cells can come through. And so again, this causes a lot more cells in this area, a lot more liquid, fluid in this area. And that's why you get this swelling. Pain you get just because of the damage to the area. And again, all of these cells in this one spot. And loss of function is kind of similar to that as well in terms of all this activity going on here. But yeah, that's what you've got. So your blood cells are leaky and more permeable. So all of these, neutrophils, macrophages, your dendritic cells, they're all kind of coming in to help out. And a lot of phagocytosis will go on. And then basically we'll talk about phagocytosis. They'll engulf these pathogens. And then this will ultimately link us to the third line of defense, so adaptive immunity. But in general, that's what we want to think about. So we can see that the blood is leaking out here because of all our immune cells coming in. So these are our phagocytes. So you can see here, it's just engulfing this little pathogen and it'll absorb it and just chop it up and kill it, basically. Okay, so we'll work through a little practice question. So which of the following matches a cell correctly with its role in an immune response? So basically working through a process of elimination here. So macrophage stimulates inflammation by secreting interferon. No, because cells that secrete interferon, it's your virally infected cells. Dendritic cell presents fragments of antigens to T helper cells. That sounds about right, because remember that dendritic cells are a really good APC, so an antigen presenting cell. Mast cell engulfs bacteria and debris. No, that would be more so a macrophage sort of thing. A mast cell, it's really key with releasing histamine. Remember that mast cells don't move, so they wouldn't make very good things to engulf bacteria. And your neutrophil secretes antibodies. I know we haven't talked about antibodies, but they're associated with the adaptive immune system, and your neutrophil is part of the innate. So that's why B is correct. Okay, so this process of phagocytosis can be seen in this diagram here. So you've got your bacteria pathogen, it comes in, it gets absorbed into this little vesicle called a phagosome. So your phagosome then fuses with this other vesicle called a lysosome. And in that lysosome, you've got all of these lysozymes, these enzymes that are involved with degrading things and chopping them up basically. So your phagosome, you've got your little vesicle with your pathogen, you've got your little vesicle with your degrading enzymes, or like digestive enzyme kind of, and then you fuse them into one, which is your phagolysosome, and then the enzymes basically get to work, and you can see that they degrade this bacteria, this pathogen, and then this is kind of just expelled. So it's quite a neat process. So this is what we can see here, phagosome, your lysosome. Step 4, you just combine them together. So that's the process of phagocytosis in general. If you are an antigen presenting cell, what will happen is you're, degrading, you're sort of like digesting this bacterium. You will get part of that, so it's antigens. And what you will do is you will go and present it on your MHC2. So your MHC2 cells, just like your MHC1, will sit on the plasma membrane. So we'll take a little fragment of this, showing your antigen, and we're going to put it on our MHC2 markers, which will just sit around here. We will then go and find our, T cell or whatever, and we will show them this antigen, basically. So that's the idea there. And then the adaptive immune system will kick in. So you can see that antigen presentation is the link between your innate immune system and your adaptive immune system. Perfect. Okay, so looking at our lymphatic system, so this is where we're getting into our sort of adaptive stuff here. So the lymphatic system, basically it transports lymph around the body, and lymph is sort of like a filter, filters your blood and all that sort of stuff, and gets rid of like fluid and all that sort of thing there. So it is like kind of closely linked with the circulatory system, but it's not blood, so it's not pumped by the heart. It just moves based on sort of like valves and your muscles moving and kind of shooting the liquid up. So that's your lymphatic system in general. Your lymphatic organs are important because they're where our cells can sort of reside or grow. So lymphocytes will sit in your lymph node. So with this process here of your antigen presentation, when you present it to a lymphocyte, you're going to be moving to a lymph node. So in terms of their names, T cells mature in the thymus, B cells mature in the bone marrow. So all of your lymphocytes start in the bone marrow. Your T cells will move to the thymus, and that's where they'll mature. Your B cells will stay in the bone marrow. That's when they mature. Hence T cells for thymus, B cells for bone marrow. And your spleen is kind of involved in this as well. Okay. So now we're getting into this real link here. So you've got your APC, say you've got a dendritic cell, we've had this inflammatory response. We've got an antigen, we're putting it on our MHC2, and we're now going to go all the way to a lymph node. So it might be in like your neck, maybe in your armpit, wherever. But we go to a lymph node, and this is where we're going to interact with a lot of our lymphocytes. So our T cells, our B cells, all that stuff hanging around there. So once we've got our T cells, again, we're getting higher up here in our hierarchy of immune cells. We need basically a more effective system to kick in. So what your adaptive T cells, your adaptive cells in general will do is they are very specific. So that's why we've got an antigen. Before we had, our little one size fits all. We don't care if it's a bacteria, if it's a virus, if it's whatever. Now we've phagocytosed this pathogen, and we are finding its specific antigen. Remember, an antigen is like a name tag. So now we're saying, okay, this is the Salmonella bacteria, this is E. coli, this is, strep, whatever it is. And we're giving this name tag, and we're showing this name tag to the T helper cell. And the, macrophage or the dendritic cell is saying, the innate cells, we've tried our best, but we need something more targeted here. That's the sort of idea that we're going with. So you can see that here. You don't need to worry about the co-stimulatory ligand. This is what you're really thinking about. So we've phagocytosed this. We've taken some of this antigen, put it on our MHC class 2, and now we're presenting it to the T helper cell. And this T helper cell is specific to this antigen. So this is another thing as well. We're not presenting this to any random T helper cell that we bump into. We're presenting, let's say this is for like E. coli or something like that. We're presenting this to a T helper cell that is specific for E. coli. So this macrophage will be in a lymph node, and it'll take it to a billion different T cells. It'll take it to the COVID T cell. It'll take it to the chickenpox T cell. It'll take it to the, tetanus T cell, whatever. It'll take it to all these different T cells. But remember the whole theme of bio, complementary. We're thinking of receptors. So we're finding a T cell that has a receptor that is complementary to this specific antigen. That is a really, really important thing to understand. Okay, so these are all our main players in our adaptive immunity. So don't really worry about naive and stuff like that. But you've got your B cell. So again, B cell will have sort of a similar receptor like this. It's to a specific antigen. If it's naive, it means it hasn't come across it yet. So in our bodies, in 2018, we had our little B cells. Oh, maybe B cells are a bad one for COVID. But let's just say, for example, right? It may not be accurate. But we've got our little B cell that's hanging around that's specific to COVID or whatever other infection. That would be naive because it hasn't come across its antigen yet. Then COVID came into our body and this B cell was saying, woohoo, I found my complementary sort of antigen. So that's the idea of when they become like a mature B cell or an activated B cell. Plasma cells and memory B cells fall under this division of B cells. I think I'll probably explain it in a diagram, but this is a good table to refer to. Plasma cells associate with your antibodies. Memory B cell, you just associate it with being your memory. So they just kind of hang around and they're good for next infections. Helper T cell, like super important, basically your coordinator of the whole adaptive immune response. They activate B cells, they activate other helper T cells, and they activate your cytotoxic T cells. Your cytotoxic T cells, remember I said were a little bit similar to your natural killer cells. They also use perforins and granzymes to induce apoptosis in infected cells. But instead of detecting when the windows are down, remember that this is part of the specific immune system. So they detect when the specific antigen is being presented on the MHC1. So they're looking in the window, and they're not only looking in the window and saying, oh, there's an invader. They're looking in the window and saying, this is the invader's first name, their last name, their date of birth, all that stuff. They're being really specific. Okay, so this is a nice diagram that sort of encapsulates this. So this is our link from our innate immunity. So we've got our antigen presenting cell here. We've got our antigen. B cells can also be antigen presenting cells. We just don't really like VCA doesn't really use it in this context very much, but they can be. So whatever the case is, often it's an APC, they present our antigen. This T helper cell will now be activated. And what it'll do is it'll make more T helper cells. It'll, what we call clonal expansion is an important thing to understand for all of these. So remember that all of these have a really specific receptor, right? So there's not a lot of them. Like, let's think about the COVID one, right? Before COVID came along, it's not really being used that much. So it would be useless for you to have like 1,000 of these cells, right? So you may have a couple, but once it's been activated, once this T helper cell specific to COVID, you know, in 2019 or 2020, came into contact with this virus, it obviously needs a bit more help, right? So it will go through this thing called clonal expansion. It'll basically just replicate and replicate and replicate. So now we have a lot of T helper cells specific to this COVID antigen. That will then activate your cytotoxic T cells, which is what we've got here. This will also activate your B cells. And the same thing will happen. Clonal expansion, you'll get heaps and heaps and heaps of these cells. So B cells will go into your plasma cells and your memory B cells. So the role of your plasma cells is just to make antibodies. And again, you've guessed it, your antibodies have those specific receptors to that specific antigen. So these are really good for like bacteria and targeting sort of extracellular pathogens. Your cytotoxic T cells, you know, we've talked about like the windows, they're really good for intracellular pathogens and like your cancers and stuff like that. So that's the idea there. Okay, so summarizing this. So we've got a lymph node, our T helper cell is activated. This is going to say, okay, let me find a B cell that has the exact same receptor. I found it, now you're activated. Now this B cell, it's been switched on, it's going to divide and it's going to create so many more B cells, and they're going to be both memory B cells and your plasma cells. So remember, your plasma cells work to produce your antibodies, and those will help kill your bacteria. Your memory B cells, their role is basically just to hang around. And then once this infection is over, the next time that we get infected with COVID, let's say, we've got a lot of memory B cells hanging around. It's their job to divide into more B cells, basically. Okay, so yeah, plasma B cells, antibodies. That's the idea there. So this is ultimately what we can see in terms of our sort of initial infection. And this is your like antigen presenting cells. You've got your T cells and your B cells being activated. You've got your plasma cells and your B cells, and then your plasma cells producing your antibodies, which look like these little Ys. Okay, I am mindful of time, so I'll keep kind of going through it. So T cell responses are targeted towards our intracellular pathogens. So again, think of our viruses, our cancers, stuff like that. So you've got you're at a lymph node, your APCs come along, T helper cells activated. That activates itself really, and it divides and it creates your memory T cells and your T helper cells. Memory T cells, exactly the same. I will say VCAR always talks about memory B cells. They never really talk about memory T cells, but it's just they have the same job. And then your cytotoxic T cells will obviously proliferate as well. So here is how cytotoxic T cells work. Again, instead of your natural killer cells detecting when MHC1 is not there, what they do is cytotoxic T cells go and they zoom in on your MHC1, and they will see that if there are any basically non-self antigens being presented on the MHC1, and then they will release perforins and granzymes and kill it. So if you've got a virus, your window is open, you're showing your viral particles or peptides on your MHC1, your cytotoxic T cell is going to come along. And remember, I can't emphasize it enough, it's all about being complementary, having the same receptor. So this cytotoxic T cell comes along and its receptor, whatever the shape is, fits really well over this viral antigen. So this is sitting in your MHC1. This is your cytotoxic T cells receptor. It's going to come along and go, this is the COVID, SARS-CoV-2, like whatever. This is the exact specific antigen that I'm looking for. I'm now going to release perforins and granzymes, and I'm going to like destroy this house. Basically, I'm going to kill this cell and kill the virus in it. So that is the idea there. Okay, so allergens are similar to pathogens in an allergic response, but obviously it's this idea of people with allergies and our response to allergens, it's a hypersensitivity. So, you've got things like pollen, dust, peanuts, compared to, if you think about a virus or a bacteria, they can cause a lot of damage, they can kill you, right? So it's valid that the immune system wants to mount a response against them. With allergens, our immune system is basically very hyperactive, and it sees these pollen, these dust, this peanut, which it isn't a source of a threat for us, but for some reason it sees it as a threat, and then it basically initiates an immune response. So a lot of this stuff is quite similar, but it's a thing that you have to realize with the allergic response is that you have this sort of priming. So what happens is if we've got someone that's allergic to pollen, so the first time pollen is going to enter their body, and we've got our immune system kicking in. So, the same thing that happened with our clonal expansion, all that sort of stuff will be specific to pollen. And we're going to make antibodies to the pollen, right? Because we think it's dangerous. But something that we do is that with these antibodies, we don't really use it to like kill it, I suppose. What we do is we take these antibodies, so they're a specific form of antibodies, IgE, and we sit them on mast cells. So that's obviously a bit different to what you do, like if it's a bacteria. So the first time your body has ever come into contact with pollen, we mount an immune response, specific antibodies are formed. These antibodies go and sit on our mast cells, and that's that. The person doesn't get sick, nothing happens. The second time pollen comes in, that's when we get this full-blown response. So the second time your pollen comes in, and this is this idea of like sort of priming, your IgE, so your antibodies are sitting on your mast cells and they're just waiting. And so as that pollen comes in, what it does is it binds to that IgE, and that causes a really huge response in your mast cells, and histamine is released, and then you have this big inflammatory response. And that's why people with allergies, have inflammation. They've got they get red, throat closes up, snotty, that sort of thing. So hopefully that makes sense. And that's kind of what I was talking about in terms of hypersensitivity. But yeah, the main thing to realize is that we've got our first exposure, like nothing happens to the outside. You don't have that allergic response. But your mast cells are just covered in this IgE. And then the second time it binds to the IgE, and then it causes that response. Okay, I'm going to probably race through this question. So cells of the immune system have different kinds of structures on the surfaces. So you've got self-antigens, receptors for self-antigens, and then receptors for foreign antigens. So hopefully that makes sense. So you've got a self-antigen. So you can see that they've all got three things. So you've got a self-antigen, you've got a receptor for self-antigens, and you've got a receptor for foreign antigens. So from the information, what are we able to conclude? again, if you want, you can take like a screenshot of it and have time to think about it yourself and stuff like that. But I will move through it. If you want to go back to it on the slides, then I recommend that you do. Um, the way that you can look at this is if you realize that you've got a self antigen, a receptor for the self antigen, and a receptor for a foreign antigen, you can basically figure out which one is which on each of these, because two of them will be complementary because you have a self antigen and a receptor for a self antigen. So they must be complementary, right? And then the third one should be a random receptor for foreign antigens. So what the question is asking you is, what is a self antigen for each of these cells? So if we go to cell P, this fits into that. So this must be the receptor and the self antigen. So this will be the foreign one. So therefore A can't be right. B, so cell R. So this fits into this. So this must be your self antigen and your receptor. This is your foreign one. Oh, yep, that's looking pretty right. Let's check the others. Cell Q. So this fits into this. This must be your self antigen. This must be your receptor. This is your foreign one, which is not right here. And lastly, this fits into this. So this must be the receptor and the self antigen. This must be the foreign one, which again is incorrect. So the only one that's right is B. I hope that made sense. Feel free to leave a comment in the chat if that didn't work. I know I went through that quite quickly. But again, take time to go back to it using the slides if you want. Sorry, I just know we have a lot to get through. Okay, so now looking at some disease challenges. So that is basically the simple, oh, it's not that simple, the process, the basic process of your first line, your second line, and your third line. So that's how immunity works in general. Now we look at, what are the types of immunity, looking at vaccination, stuff when immunity might, be going wrong, all that sort of thing. Okay, this is probably the easiest part of immunity, I would say, maybe. So you've got your natural and official immunity that you have to distinguish between, and your active and your passive immunity that you have to distinguish as well. So your natural immunity, this is when you have ultimately antibodies that you've made yourself, basically. So with your natural immunity, you have basically been infected by something and then you are expressing antibodies. In your artificial immunity, this is when the antibodies are basically not your own, ultimately. So with your active immunity, this is when your antibodies are produced due to the exposure of a pathogen. So again, this sort of, again, it might not be a natural process of infection, but this idea that your body is making its antibodies itself. And then with your passive immunity, this is when you are not making your antibodies yourself. So it's all about antibodies. So natural immunity, the antibodies have occurred due to a natural process. Artificial immunity, the antibodies have occurred due to an artificial process. Active immunity, the antibodies have occurred due to your body sort of making them. Passive immunity, they've ultimately been made by something or someone else. So a really common example that they use is in terms of your natural immunity, you think about your natural active is like your typical thing that we looked at. So you're infected by a pathogen and you mount a response, you have antibodies as a result. That's your natural active immunity. Your natural passive immunity is when you basically are given antibodies through breastfeeding or through your mother that way. Because it's natural, because it happened without medical intervention, but it's passive because the antibodies have been made by your mother and not by yourself. In terms of artificial, so artificial active is your vaccination. So again, very similar to to natural active. The body's mounting its own immune response, you're making your own antibodies, but it's just the source. Natural is I bumped into someone on the street who had COVID. Artificial is I got the vaccine. And then lastly, your artificial passive. So you are getting someone else's or something else's antibodies via medical intervention. So if you are bitten by a snake or something, then you will get this antivenom, which contains these sort of like pre-made antibodies. So that's why it's passive, because you're not making the antibodies yourself, and it's artificial because they're being, it's not coming naturally through your mum, it's coming from wherever else. Okay, Hopefully that made sense. So in terms of our in terms of our vaccinations, so the process with this is kind of what I've just described in the previous slide. So we um work to kind of get immunity by injecting a little part of a pathogen into ourselves. So we saw this whole process of when, you've got your APC and you get your clonal expansion, all of that. So we want the end results of antibodies, but we don't want the initial result of getting sick, right? So that's why we use an inactivated form of the virus or an attenuated or a weakened form. So, yeah, so basically like the pathogen is still alive, but it's just very weak or inactivated in terms of like it can't cause disease at all. So that's why when you get a vaccine, you don't usually get super sick. So that's why you insert part of that pathogen, and the idea is that you have inserted the antigens basically. So on this inactivated or attenuated form of the virus, let's say, you've got a pathogen. No, sorry, antigen. You've got an antigen from that pathogen, and then your body's going to mount a response against that. So then again, the same thing's going to happen. Your T cell is going to be activated with that specific receptor, your B cells, plasma cells, so on and so forth. And so the really important thing is you get those memory cells. The memory cells are really, really important because that means, say like I've never gotten, I don't know, chicken pox or something like that. So I've never gotten there's a chicken pox vaccine. So I say I've never gotten chicken pox. So I get this chicken pox vaccine, and now I've got this whole system going on, and now I've got memory cells to chicken pox. What happens is when I encounter chicken pox in my day-to-day life, instead of getting sick because my body, hasn't seen chicken pox for the first time and it takes a long time, because I've got these memory cells hanging around from my vaccination, my body will recognize that chickenpox antigen and it'll mount a really huge response. And it means that I won't get sick because it's going to work so fast. And that's kind of the basis of vaccinations. So the really important aspect of that is having those memory cells. Okay, hopefully that makes sense. So this is a nice diagram to represent that. So this is your initial exposure. So I'm getting the vaccine for chickenpox. So my antibodies are being made, my memory cells are hanging around, all that sort of stuff. And I'm getting this sort of primary immune response. So I've gotten a nice amount of antibodies. You can see that this takes a long time. So let's say that if I'm not it's not through vaccination, it's through actually being exposed to chickenpox. This is a long time to be sick until your body actually gets to work. Anyway, so I've got my antibodies, they're hanging around. The numbers dwindle a little bit, right? And then, chickenpox is gone. The antibody number is pretty low, but I've got these memory cells still hanging around. Suddenly, someone else is infected by chickenpox. I bump into them. Those memory cells are going to say, I remember you. I'm going to go through my little clonal expansion thing again, and I'm going to make all of these new plasma cells and all of this sort of stuff. And you can see that the amount of antibodies skyrockets. And that's because, remember at your initial exposure, maybe you only had 1 or 2 cells that had this specific receptor to this chickenpox antigen. But now I've got a bunch of memory cells hanging around after this, vaccination or after this exposure. So now I've got heaps of cells that are specific to this. So once this chickenpox virus enters my body for the second time, the chance of it bumping into something that has a specific receptor is much higher, which is why I get this really quick response. And because there are so many other chickenpox, memory cells hanging about, once I'm activated, I activate all of them, and then you get heaps and heaps more cells. And that's why you get a much larger response the second time as opposed to the first time. And then it just goes on and goes on and goes on. And this is the process of when you get a booster. So if you had your COVID vaccine, you get this initial response. And then if you get your second vaccine, you get a nice bigger response and then a booster and so on and so forth. So you have a nice high level of immunity hanging around. With COVID, obviously it's a bit different because it is a virus that mutates pretty frequently. If it is a bacterial infection or a viral infection that doesn't mutate that much, then again, it's like pretty fine. You can have 1 vaccine or 2 vaccines and or one vaccine and a booster. But that's why, influenza, you have to get that every single year because it mutates. And so the immunity that this is the immunity that I've got for the 2022 influenza. And that's great. So like if that comes through, hopefully I won't get sick. But the 2023 one is completely different. The antigen looks different. So then that's why you have to get another vaccine every year. Okay, please let me know if that didn't make sense. Just pop any questions into the chat. The main principle with your vaccinations and for them to work is this idea of herd immunity. So herd immunity is when you basically have enough protection from everyone being vaccinated that your vulnerable members of the community aren't going to get sick. So the idea here is you generally need a pretty high number, so about like 95%. And then that's when you've got this sort of scenario going on. So initially, nobody's immunized. These people in red are sick. Everybody around them is susceptible. They get them all sick. Everyone gets sick. So we've got a couple members of the population who are immunized. Again, these people are sick. The people who are immunized, they're not going to be sick, right? Because they're immune to the disease. But again, we've still got quite a lot of people getting sick just based on proximity and contact. If we've got most of the population immunized, we can see that this, metaphorically, creates a nice barrier that prevents these people from not getting sick. And so these people may be people like the elderly or the immunosuppressed who perhaps can't get vaccines or who would, be sick if they were to get a vaccine. And that's what this idea of herd immunity is. It's very much in the name. It's the protection that the rest of the population gives to those who aren't able to be vaccinated due to the rest of the population being vaccinated. So hopefully that makes sense. And it's obviously very helpful in containing and almost like eradicating diseases as well. Okay, so moving on to immunotherapy. So this is when we basically influence the immune system or manipulate the immune system in order to cure certain diseases. So monoclonal antibodies are a specific example you need to know. You need to know them in the context of cancer and autoimmune diseases. So with our monoclonal antibodies, we have, just like any other antibodies, and they are specific to, for example, let's say cancer. So what we do is we get basically a cancerous cell and we figure out an antigen that a cancerous cell may have, and we put this into like a mouse. For example, these are monoclonal antibodies that are made by mice. So we put them into a mouse, and then what we do is we put them in a mouse. The mouse will mount an immune response against this specific antigen. So, again, think about that little diagram with everything, and they'll all have this specific receptor to this antibody. So we're going to create plasma cells, and they're going to create antibodies that have specific receptors and will target this cancerous antigen. So the idea of epitopes here, so we have antibodies, and then they're obviously specific to antigens. Antigens themselves have little parts of them. So maybe I'll see if I can draw this out. Okay, so let's say that this is a bacteria, and we'll say that this is an antigen. Now there are specific parts of the antigen that antibodies will bind to. So you might have this sort of sitting on here. Obviously this is a very, they're molecules and whatnot. Okay, I don't even know. But yeah, right? So we've got 3 different things. So these are what we would call epitopes. So you can see that things can be specific for an antigen, but then they can be even more specific to certain epitopes on that antigen. So that's why you can have, for example, here, you can have 3 different antibodies for one antigen. So that's the idea there. So some antibodies will be more effective for the epitopes than others. And so when we figure out what antibody is the most effective, that's the one that we'll harvest from that mouse, and we'll clone it, and then we'll use that in that individual, because we know that that's a really good antibody. So that's why it's called like monoclonal, because it's specific to one particular epitope. So if that makes sense. So you just put it in the mouse creates these plasma cells, you take the plasma cells, you make lots of them, monoclonal antibodies. Okay. So this is a nice little diagram of that here. So this idea of we pop it in a mouse, it does its thing, and we get these antibodies. So we fuse it with these tumor cells. And that means that the plasma cell will just keep on going and going and going and going and going, because they basically, we choose these specific cells that don't have like an end date, basically. And that means we can produce even more and more and more, because the goal is just we want to create as many antibodies as we can. So obviously this is quite useful. It is a developing area of research. There's lots of other diseases that it's being used for. Again, you have to know it in the context of autoimmune diseases. So like rheumatoid arthritis, for example, there's a monoclonal antibody that's really popular. It slips my mind now. It's like rituximab or something like that. I don't know. You can have a look into it. But yeah, that's being used, in the context of autoimmune diseases. So you can use that if you've got an autoimmune disease. So sometimes your T cells, they may be targeting your self cells because there's something a bit dodgy with the T cells going on, right? So you can use these antibodies to actually target those T cells as opposed to, your cancerous antigens. So that's another way of using monoclonal antibodies. But they're really helpful because they're very specific. As you guys know, I've been using the word specific about 1 billion times. Again, use it in all your short answers. It comes up a lot in bio. But yeah, it's very specific. And compared to, chemotherapy and radiation, which just target fast replicating cells in general, it can result in less severe side effects because they're more targeted to cancerous cells as opposed to, like hair cells or skin cells or stomach cells, stuff like that. Yeah, that's basically the idea there. Okay, so in terms of pathogens, needing to understand, it's just kind of this context of COVID, really. This idea of when known pathogens, which you sort of eradicate, can come back. So it's just this idea of, especially it says there in the study design dot point, in a globally connected world, you've got people moving about to different locations, and we move very rapidly now. So it's very easy to be, like in Australia, where we've got a lot of diseases that are eradicated, and you may go to another area, and then, pick up a disease that's endemic there and then bring it back. And that idea of, again, coming into contact with other people who travel, and that can spread. And so that's this idea of reemergence of pathogens that we know and are aware of, but that we've kind of eliminated. So that idea of them kind of reemerging. That's why sometimes you get like measles outbreaks, or, like tuberculosis outbreaks and all those sorts of things. And yeah, it may be from areas where tuberculosis or malaria is more endemic, and then that idea of kind of bringing them back. The other thing you need to be aware of is emergence of new pathogens. So this is obviously linking quite closely to COVID. It's this idea that nobody's ever seen it before, and it's this emergence of a new pathogen. And it's this idea that these pathogens are more likely to cause pandemics. So things like, malaria, tuberculosis, they're endemic in certain areas of the world, but they're probably less likely to cause pandemics because we are very aware of them. Whereas, as we've seen with COVID, if you've got something that's quite new, and particularly something like COVID that's quite fastly mutating, it's very hard to get a grip on it early. And then that idea of nobody having prior immunity, nobody has your little memory cells hanging around. So then that's when you get these kind of pandemics, and new pathogens can do a lot more damage, is the idea there. Okay, so thinking about the effects of European settlement in Australia and the effect on the Aboriginal and Torres Strait Islander population, the idea here is kind of what I was mentioning before in not having any immunity, not having any prior exposure. So it's this idea that in Britain, like before they came to Australia, we're bringing a lot of communicable diseases with us on the boat, right? And it's things that are endemic to this area of Britain, for example, and that these people have been exposed to. So they're bringing that with them. And we're then bringing it into a community that has not been exposed to these viruses before. So we can see, smallpox, measles, influenza, this sort of thing that again would be prevalent in like the UK or in Britain. And then it's this idea of bringing it to an area where Aboriginal and Torres Strait Islander people hadn't been exposed to before. So they don't have any prior immunity, and they're extremely susceptible to these diseases, and it can wreak a lot of havoc. And so that's what we saw with smallpox, this idea that there was an epidemic. There's lots of areas where you had really large outbreaks because ultimately nobody had immunity to it. Whereas, some of the other people, like other people in Britain and obviously those that colonized Australia, they would have certain levels of immunity to it, having probably been exposed to it in childhood and things like that. So that's just one of, the many obviously negative effects. And this idea of, globally connected worlds and things like that, and this spread of travel, and how that can bring diseases. And we see that in lots of other examples of colonization as well, and how that can really wipe out and ravage a lot of communities. So hopefully that makes sense. It's just this idea of being exposed to something that you haven't been exposed to before and being extremely vulnerable to it. Okay, so in terms of the transmission of infection, so we're looking at this idea of this chain of infection. So it's this idea that when you have a pathogen, it kind of follows all these little steps. So you've got an infectious pathogen, you have a susceptible host, really, and this pathogen obviously infects this host via this portal of entry. In terms of these reservoirs, it's where the infectious pathogen basically kind of grows and just sits, and it then exits that. It's then transmitted into somebody else. So the reservoir may be another person, really. So the reservoir may be, another person, and then they're able to exit that individual, and then it's transmitted into somebody else via that portal of entry. Hopefully that's making sense. And then you've got the host who's infected, and now they're creating more pathogens. So hopefully that makes sense. You guys should be really on top of this by now with COVID in terms of ways of controlling transmission, particularly of our communicable diseases. So hand washing, physical distancing, face masks, all the stuff that I'm sure you guys are really, really aware of. So there's a lot of modes of transmission. So respiratory transmission, droplet transmission. Respiratory transmission is basically via the respiratory system. So you kind of hack it out via your respiratory system, and then it goes into somebody else's respiratory system. Droplet transmission is kind of similar, but a little bit different in that the pathogen can basically travel in the air via droplets. So when you sneeze on something and you then like touch it and then touch your face or whatever, that idea of being transmitted via droplets. Contact transmission, so via direct contact. So for example, the fecal oral route. So yeah, if the pathogen is excreted in the feces and somehow that kind of gets, you may not wash your hands properly after, going to the bathroom, and then you may touch your mouth, or, you may eat something, and then that gets in via the oral route. So that idea of fecal oral. Vector transmission. So we talked about malaria at the start, this idea of the pathogen often being like a little parasite in that vector, and then that gets transmitted as well. So if you think about malaria, for example, that idea of avoiding the vector, preventing areas, so like preventing being bitten by a mosquito, getting rid of like mosquito breeding grounds, that sort of thing. And then sexual transmission, so kind of bodily fluids, that sort of thing, pathogens spreading by there, so like your STDs and all that sort of stuff. Okay, so here is a bit of a question. We'll try and read it really fast. So BSE is a prion disease of cattle. So again, how we were talking about how prions often commonly spread from animals to humans. So it's sometimes called mad cow disease, caused by feeding cattle food that contains prions from other infected animals. So you've got prions from other animals, and then you feed that to cows, and then basically like the humans eat the cows. Blah, 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 blah. The time between infection and symptoms appearing can be up to 5 years. So you can have a prion in you, you can eat infected meat, and then 5 years later you get the disease. If you think about it, that's an extremely long incubation period. And you can imagine the kind of trouble with that. There are concerns that VCJD in humans could be caused by eating infected cattle meat. So this is kind of a variant from that. So mad cow disease is in your cattle, like the disease is in the cow, hence mad cow. And then when this kind of spreads to humans, it's your Creutzfeldt-Jakob disease. So yellow fever is a viral disease that affects humans. So here we're talking about something different. So this is a prion, this is a virus. Yellow fever is a viral disease that affects humans. The yellow fever virus can cause symptoms 3 to 6 days after infection. The virus is carried by a mosquito vector. Which combination of approaches would be most effective at controlling the risk of outbreaks of both VCJD and yellow fever? So you can see how there's like so much information, and obviously it's helpful for context, but if you break it down, again, you're not expected to know what VCJD is, you're not expected to know what yellow fever is. Whittle this question down. It's asking, how can we prevent the infection of this prion? How can we affect an infection of a viral disease via a vector? That's the idea. They could have swapped the word VCJD and yellow fever out for anything in the world. Again, you're just having to apply knowledge that you have already. So, prevent all cattle that show symptoms of mad cow disease from reproducing. Remove breeding grounds for mosquitoes. Test all cattle for the presence of prions. Ensure that all healthcare professionals wear gloves when working with infected patients. In terms of yellow fever, so it's a viral disease that affects humans. Virus is carried by a mosquito vector. So you're going to want to look for something that is particularly targeting mosquitoes. So removing breeding grounds for mosquitoes sounds pretty good. Ensure that all healthcare professionals wear gloves when working with infected patients. Probably not the best thing. Ensure that people take measures to reduce their chances of being bitten by mosquitoes. Yep, that's pretty good. Instruct people who are infected with yellow fever to wear masks in public places. Not the best thing, because this would be effective for maybe respiratory transmission or droplet transmission, but not really for something that's caused by a mosquito. So you're kind of down to A and C. So you've got prevent all cattle that show symptoms of mad cow disease from reproducing. Destroy all cattle that have been fed infected food containing the prions. So again, we're thinking of A and D. They could both be quite helpful. So again, what this comes down to is what is the best answer. So if you read, A or C sort of by itself, you may be inclined to choose them, which is important. Oh my gosh, which is why it's therefore important to read all of the answers so that you can get the best option. So prevent all cattle that show symptoms of mad cow disease from reproducing. So that sounds, relatively good, but think about the method of transmission. So prions from other infected animals. So basically the cow's got the prion in it, right? And then it becomes VCJD because somebody eats that cow. So if you prevent all cattle that show symptoms of mad cow disease from reproducing, okay, that might be good, but does that stop that cow from having mad cow disease? No. Does that stop that cow from being cut up and then given as food? No. So that's why C is the better option. Destroy all cattle that have been fed infected food containing the prions. Because of that idea that you're preventing at all costs that from becoming meat that someone will eat. Whereas A, prevent all cattle that show symptoms of mad cow disease from reproducing. That cow can still become meat and someone can eat it and they can get BCJD. Hopefully that makes sense. Okay, I know we kind of raced through that one, but hopefully that's all making sense. So we'll finish off with, experimental design in our exam study. Again, the study design is really helpful. With this, you just kind of have to fish through it and see what's actually useful, what's not. Most of the important terms are in here. When we get to it, I will talk about reproducibility and repeatability because that's something that's been introduced with this study design. But super, 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 super important. If you haven't done your area of study 3 SAC yet, do all of this stuff as soon as you can because it's really important for that SAC, but also for your exam. There are always questions on it, particularly towards the end of the exam. I want to say towards the end of the exam is when your questions get longer. So that's when you start to get big paragraphs, when you start to get lots of multi-part questions, and it's sort of where they try, or not where they try, but where they do tend to bring in a lot of area study 3 stuff. I remember when I did bio, I hated it. I hated it with a passion. Yeah, that's basically it. But you just have to study it, and eventually, you know, you might find a liking for it somewhere. I personally didn't really, but it's really important. And I think because I hated it, I never wanted to study it, but I had to. Otherwise, I would have lost a billion marks. So just be mindful. And I think something as well, I used to tell myself, like, oh, I've done this for lots of years. Like, I know it really well. It's fine. But you have to work, like, doing practice questions with it, so that you have ideas in your head of how VCA will ask questions relating to photosynthesis, relating to cellular respiration, relating to transcription, translation, DNA manipulation, immunology, all that sort of stuff, and how they will apply it in that context. You can't just say, like, oh, it's an application question, so I'm just going to apply it when I get there. It's an application question, so I need to practice applying it in as many different scenarios as I can, is what your thinking should sort of be. Okay, so knowing your variables. So your IV is the thing that you manipulate. Your DV is the thing that is being measured. The whole point of your experiment is to figure out what the effect is of the IV on the DV. Your controlled variables are basically any variables that could affect the DV that you obviously don't want to. They need to be kept controlled. If you do not control these, there is no way of saying the IV has caused this change in the DV because it could be this other variable that you haven't left controlled, and then your experiment is useless, basically. So your experimental group and your control group, very important to distinguish controlled variables from your control group. Controlled variables are the things that could be the IV, just the little variables. Control group is the whole, like, setting. So your experimental group is what is exposed to the IV. So if you are observing light intensity and its effect on photosynthesis, your experimental groups will be, I don't know, dim light, average light, bright light. Your control group will be something in the dark. It's something that is not exposed to the independent variable, and it acts as a baseline comparison, and it also makes sure that you don't have any confounding variables. So you are checking light intensity on photosynthesis. You've got your dim light, your mid light, and then your bright light. And then, your dim light doesn't grow that much. Mid light grows a little bit more. Your bright light grows amazingly. If your plant that is in the dark, if that grows amazingly, you're kind of thinking, something's going wrong here, right? Because we're not exposing this to the independent variable. So it should serve as a baseline. It should be like a 0, really. Or it should, if it grows 3 centimeters, then you'd expect everything else to grow above 3 centimeters. It's that idea of that baseline comparison. So that is why the control group is really, really important. And whenever you write about an experimental design, you need to make sure you identify that you have a control group. Okay, so your validity and your reliability. Validity is basically, is this measuring what it's meant to measure? So you can think about that in terms of equipment or in terms of your experiment. Is this experiment, have I designed it in a way that it actually tests the IV and the DV? So I want to check the effect of light intensity on the rate of photosynthesis. Is my experiment actually set up in a valid way that I am able to, like, answer the question, basically? It's similar to accuracy, but accuracy is basically, is this correct? Like, my plant has grown by 3 centimeters. Is this sort of like actually how much, it's grown by? Has it grown by 30 centimeters and I'm using the world's worst ruler? That sort of idea. Reliability is if I repeated the experiment, is the result going to be the same? Precision is, are all my results within a narrow range? In terms of your repeatability and your reproducibility. Repeatability is same time, same place. Reproducibility is different time, different person, different place. So your repeatability, again, think of the name of it as well. Repeat versus reproduce. If I'm thinking about repeatability, it's if I perform these measurements again in the exact same context, the same person, the same equipment, am I getting the right results? If it's reproducibility, is it if I'm a different person on a different day in a different environment using different equipment, am I getting similar results? So that's what you should think about as well. So having a large sample size, repeating the experiment is really important for your reliability and getting rid of your your error. It'll come up. Yeah, your random errors. That's the way I was saying. I was going to say standard errors. Your random errors. Having a large sample size and repeating is really important for that. So random errors are just that, really random errors, sort of like fluctuations in atmospheric pressure, temperature, that sort of thing. It's completely random and it will affect like one random result. Whereas your systematic errors affect every single result consistently. So it's often to do with equipment. So if you've got a measuring thing, like I said, like the world's best ruler, if you've got a ruler and it measures, it's got your 1 centimeter, 2 centimeter, 3 centimeter, but in actuality, each of those centimeters are 1.2 centimeters apart, it's going to create a systematic error because you're using it to measure everything and it's going to be out every single time. Whereas your random error would be, the temperature in the room changed. So then, I don't know, the plant wilted or something like that. So that's what you're thinking of there. So be really aware of errors and ways to improve errors and things like that. Very, very important. Qualitative and quantitative data, I'm sure you've been bored to death of this sort of thing. Qualitative, quality, descriptive words, subjective. Quantitative, more objective, more numerical. Quantitative data is what you sort of aim for. Qualitative is still very helpful, but quantitative is just a little bit more robust. Okay, so in terms of answering an experimental design question, very different to answer an experimental design question in an exam versus as you would for your like area study 3 SAC. So there are a couple of things just to kind of note. So it depends how many marks. Generally, there might be about 4 marks-ish, maybe less. So you just need to be really concise. You're not writing out, when you're writing out a method as well, you're not writing out a method as you would in your area of study 3 SAC, where it's like 1 billion steps long. You're just being really concise and giving a nice overview about your method. So your treatment and your control groups, your experimental, your control groups, remember that control group. It's really, really important. Students always forget it. Remember to include a large sample size. Remember to be as specific as you can. Dependent and independent variables, your controlled variables, again, listing about 3. A sentence or two. Nothing more, nothing less, about your method. But again, you don't have to go step by step by step by step. This is a 4-mark question, not a 40-mark question. So really be mindful of that. But at the same time, don't be too brief. You need to give a proper explanation of what you're doing. And lastly, repeating the experiment. Talk about this idea of, your reliability or your repeatability, that sort of thing. And talk about how you're minimizing your random errors through that. Hopefully you all said that at the same time. But yeah, random errors are what you're thinking about in terms of repeating that experiment. Because if you think about it, if you have like 4 measurements and one of them is affected by a random error, that can skew your results based on if you've got 40 measurements. 1 in 4 things being dodgy, that's going to create a bigger effect than 1 in 40 things. So just be really mindful of that. Okay, we will go through this a little bit. So 2 researchers have discovered that they did not properly label a bottle containing the enzyme amylase, which catalyzes the breakdown of starch. The amylase could either be amylase from a human or from Thermus aquaticus, found in 80-degree hot springs. So devise a test they could conduct on the amylase to determine its source and include in your answer which characteristic of enzymes makes this possible. So we're thinking about the optimal conditions of enzymes here in terms of temperature. So optimal in a human would be about 37-ish degrees. Optimal in this Thermus aquaticus is 80 degrees. So you're going to try and figure out what works in what environment. So you'll have about 4 groups here. You'll actually have 2 different control groups because you will have your control groups at different temperatures. So you're going to do one in your human sort of thing. So you'll have your amylase with your starch in your 37-degree temperature. And obviously if that works, and you get glucose from that, that'll tell you that it's a human. You will also have your amylase by itself at 37 degrees. That acts as a control because you don't want any glucose to come from that. Your Thermus aquaticus, the same thing. You'll have your starch and your amylase at your 80 degrees. Again, if that works, you get glucose from there. You're thinking of your Thermus aquaticus being from there. You're also going to have another control group. You're going to have your starch at your 80 degrees. So you can see what I mean by 2 control groups there. So your independent variable is essentially the temperature. Your dependent variable will be the breakdown of starch. So that kind of indication of the level of glucose there. Your controlled variables, so you'd think about the amount of amylase that you're using, the amount of starch that you're using, that sort of thing. In terms of controlled variables, in terms of your method, you just kind of talk about what we talked about in terms of that setup. So maybe you leave it for 10 minutes and you check the glucose reading every 2 minutes perhaps, and you would repeat the experiment. That's a sort of idea there. I don't know if I've missed anything, but you would just follow this little step. Okay, maybe I'll leave you to have a go at this just based on the time. But a molecular biologist suggests that the binding of a specific hormone to muscle cells causes them to produce a much greater amount of a specific protein. Design an experiment that the biologist could undertake to determine if his hypothesis is supported. So your independent variable here would be the presence of the hormone. So basically you'd have a specific hormone to muscle cells. So you'd have muscle cells and you would have some exposed to the hormone, some not, and you're measuring how much protein is produced. So dependent variable is the amount of protein X that's produced. Your independent variable is basically the presence of this endemon, and your control group is the muscle cells without endemon. You might use varying levels of endemon perhaps. But yeah, your control group would be, your controlled variables would be maybe the amount of muscle cells that you've got, the, presence of other hormones, you would want to minimize that. Perhaps, yeah, that's kind of what you're thinking of there. In terms of your method, kind of what we've talked about in terms, again, of that setup, you would just leave it for a couple of minutes, measure the protein level. Think about how you might measure the protein level. Perhaps with a little protein X detector. I guess that's the most specific thing to be using. And yeah, you would repeat the experiment as well. Okay, but again, feel free to write that out and elaborate a little bit more on that. Okay, so these are some of the ethical principles. In this study design, bioethics are emphasized a lot more than they have been in previous years. So integrity, basically just being honest, communicating all of the results, even if they're bad. Justice, ensuring that everyone is treated fairly, ensuring that, whatever you're researching, like say it's a drug or something, it'll be accessible to like all people and you're not going to create this sort of unfair market really. Beneficence. So beneficence and non-maleficence are sometimes confused. Beneficence means doing the most good and non-maleficence means doing the least bad, basically. So beneficence, you're always thinking about, promoting benefits, always doing good. And then non-maleficence, you're just wanting to avoid harm. You're wanting to not, cause any negative consequences along the way, basically. And respect, I'm sure you guys are all familiar with respect. Okay, so approaches to certain bioethical issues. So they can be consequences-based, duty-based, or virtues-based. So consequences-based basically means that the emphasis is placed on the outcome. So it doesn't matter what you did along the way, as long as the outcome is ethical. It matters what you did along the way, but as long as the outcome is ethical, that's where the most importance is placed. Duty-based or rule-based is basically this idea that whatever the outcome is, you have to be moral along your journey, or be ethical along your journey. So that's they're almost a little bit opposite duty-based and consequences-based. And then virtues-based is this idea of if you acted sort of morally. Virtues-based is kind of a bit like a bit random, but consequences-based, as long as your outcome is ethical, that's your main focus. Duty-based, as long as along the way everyone's being treated ethically, that's the most important. And virtues-based is as long as you acted really morally, that's where the most importance is. Again, this is very application-based. You'll likely get like some sort of scenario, and then Vika will say, if you were to act in use a virtues-based approach, what would this involve in that specific, scenario or context? Okay, so a new drug to treat malaria is being trialed by scientists. To apply the concept of non-maleficence to their research, the scientists should ensure that what? So non-maleficence. So any harm to the participant resulting from the trial is not disproportionate to the benefits obtained from using the new drug. Yep, that sounds pretty good. Data that shows the new drug is ineffective is not published. That would be to do with integrity, and that would be breaching integrity. Consent is obtained from all of the participants in the trial. That's just, respect, informed consent, really. The participants experience only the benefits of the trial, more so sort of beneficence in that way. So precision medicine can be used to develop anti-cancer drugs that target and silence the gene or genes that cause a particular cancer. The government does not provide funding for many of these drugs, and patients may need to spend upwards of $100,000 for one course, when many courses of the treatment are likely to be needed to prolong life. This leads to unequal access to these life-saving drugs in society. Situation shows a lack of, hopefully you all said, justice. Again, you had like 7 sentences there, or just less than. You could literally just use the last sentence. This leads to unequal access to these life-saving drugs in society. You can answer the question just based off that. So again, be mindful of not getting too caught up in unnecessary or irrelevant information. Okay, so we'll finish off with just kind of going over some exam study tips. My biggest one, practice questions. I used to hate practice questions. My teacher told me to do a lot of them, and I used to think it was silly until I literally, as I mentioned, didn't know how to answer, or realized I didn't know how to answer bio questions. I would recommend practice questions for all of your subjects. For me, bio was the most important subject that I had to do practice questions for, just because they're so picky. You can think that you're getting all the points, and low-key, you are getting all the points, but if you don't write it in a specific way, they'll mark you down. So use practice questions. Again, I know you guys are using a new study design, so some of the old exams may not be as helpful, but you can still go through them and you can still sort of recognize what is on your study design and what's not. Focusing on your weaknesses is a big thing. Okay. I don't want to lie, I didn't really do it for my VCE subjects that much, but I did it for the UCAT when I was studying for that, and it was really, really helpful. And I think I kind of take that with me in uni now, this idea of making a little log of everything that you get wrong and the types of questions that you typically get things wrong on. You should do those over and over until you get them right, really. So this idea, I kind of mentioned I would love to do questions or even subjects. I'd like to do stuff that I knew I was good at. But then that doesn't help you in the long run, because then I would stay bad at questions that I was terrible at, basically. And then so the more you practice and do those questions, then the better at them you'll get, and then you'll want to do more of them. Asking questions. So obviously your teachers, your friends, your tutors, on the internet, you can find resources for bio everywhere, because everybody studies bio, as in it's done in uni, it's done in every school overseas. That idea of bio being a very, very, very popular subject. So there's no shortage of resources. So please use them to your advantage. And also, as we get to this time of year, going into term 3, again, a lot of you may be in year 11, a lot of you may be in year 12, particularly those for in year 12, but of course, if you're in year 11 as well, you can get really stressed at this time of year. So it's really important to kind of check in with yourself. Make sure you're not burning yourself out, especially not now. Term 3 is often the one where you lose a lot of motivation. I know I did, and I think I got slightly burnt out. Yeah, just like I got really demotivated by the middle of term 3. And so it's important, I think, not to go too hard too fast and always be checking in on yourself. And something I always do is I always gave myself breaks. I knew that if I just pushed a little bit more, sure, like maybe I would have gotten an extra mark and a bit of a higher grade, but it would have come at the expense of my like mental health, really. And I knew that it wouldn't have been productive for me anyway. Again, of course, everyone's got different learning styles, but I think it's really, really important not to ignore when your body and your mind just really need a bit of a break. Okay, so in terms of your exam, I know this is only coming up in a while. We're talking about exams at this time, but don't get too psyched out. You've got a lot of time. And that's kind of what this point is relating to as well. Everything that's gone on in the past 6 months with your SACs and all that, you don't need to worry about them too much. Like, obviously your SACs are important, try your best, but your exam is where most of the money is. And there's no reason why you can't improve on this semester's SACs and do well in your exam, and then that can take your grade up a lot as well. My teacher used to always say that SACs are a draft. So they make up, a smaller percentage of a bigger score. So it's important not to focus and dwell on SAC scores too much, especially when you've still got a whole half of a year to focus on. It's really important. And don't think and don't get into, the sort of headspace that, I've done badly in my SAC, so I can't do well. Even if you do badly in all of your SACs, which I'm sure none of you will, but, moving closer to the exam, still try in your exam. You can still do well. It's a lot more, a bigger proportion of your grade. So just always, keep doing your best and don't ever think that a bad SAC will mean a bad study score, because that's not the case at all. I think in bio, I had a SAC that was really quite low. And I remember I thought the same thing, like, oh, okay, I already flunked it. But then I didn't. So the same can happen to you. Learn from your SACs. I know my teachers are a bit funny in like giving SACs out, but sometimes they'll let you like look at it, in there. Like they might not let you take it home, but hopefully they'll let you read over it. And just learn. It's just the one thing, like learn from all of your mistakes. Like when you get into your practice exams as well, note your mistakes down, note especially ones that keep on recurring and why you made the mistake. I always say marking your exam is almost more important than actually doing it, because there's no point in doing an exam and not marking it, because then you're going to make those same mistakes, a billion times. Really, really, really try to learn from things that you get wrong and also things that you get right. Even when you get things right, think, did I get this right? Like particularly for multiple choice, did I get this right because I, came to the right conclusion via the right pathway? Or did I just kind of like guess a little bit? Or did I think something completely wrong and I just happened to get this one right by chance? So yeah, just be aware of that. Unit 3, I feel like I mentioned this before, but keep on revising your stuff for Unit 3. Do not let it slip to the back of your head. Now is a really good time to think of all the stuff from Area Study 1. I know most of you would have just finished like your, if you haven't started immunity already before the term ended, but you might have finished, your cellular respiration and your photosynthesis. Don't forget your transcription, translation stuff, your protein stuff. Super important. With antibodies and immunology, antibodies are an example of proteins. So all of the protein stuff from Area Study 1, that's all relevant still in immunity. Anything that you talk about with proteins can still apply in terms of antibodies. So, always continue to revise your Unit 3 stuff, and it just saves you time, because otherwise you're going to get to September, October, you're going to have to go back and relearn everything, and it's just going to be a lot harder than if you just did like a couple questions on the weekend, or the week, or, whenever you want, but just like slowly revising stuff and keeping it in the back of your head rather than losing all of it, and then having to, catch up again a little bit later. Okay, practice exams. Emphasis on maybe time. I personally started practice exams a little bit later, I would say, like um maybe like 6 weeks out, I want to say. Take what I say with a grain of salt. It's been a long time, but yeah, I would say like 6 weeks out. I know some, the other tutors and some other students, some of my other friends even tend to start practice exams a lot earlier. It just depends on where your confidence levels are at, just depends on how you feel you wish to prepare. For me, I didn't like the idea of doing practice exams before I was really confident with everything. Practice questions are different. Um, but practice exams, I preferred to be a little bit more on top of the content. So I would actually be sort of like mimicking how I would feel, on the day. And I feel like if I did practice exams earlier, like practice exams before I finished the content, a lot of my friends did that. A lot of my friends did that. I just never really liked that idea. But again, it's whatever works for you. But yeah, with bio past exams, so many of them are accessible. Again, there's heaps of VCAR ones. Unfortunately for you, you're just gonna have to like sift through what is still relevant on this study design. But yeah, a lot of schools do those like exam booklets, and so those are quite helpful as well, and they tend to give them out pretty early. Um, but yeah, use your reading time. Think about how you're going to approach it. Are you going to do short answer first? Are you going to do multiple choice first? Figure out that sort of stuff. And yeah, try to build up your stamina as well. So do it under timed conditions, obviously doing it closed book, and mark your exams, of course. Don't waste your practice exams is kind of what I think, because otherwise you could just be doing practice questions. We kind of talked about this in terms of keeping track of your mistakes, but yeah, it can be really helpful to observe them. Okay, I think this is the last slide. So we've talked about complementary, this idea of being specific just comes up in bio all the time. Like I'm almost sick of saying it so much. All the time. So just be aware of it and make sure you are explaining things, especially with immunity, like on and in enough depth, but also concise. I know that's so annoying to hear that. Like use a lot of words, but don't use any. But being concise in bio is really important, especially because it's just so much stuff. Like I tend to be a waffler when I write. And I remember like I was thinking about this one question in my exam that I did in 2020. And I remember I like waffled on. And as I was writing it, I remember like I used extra line space and I was writing in the corners and I was like, oh, as I wrote it, I like knew like, yeah, okay, like I've done what I'm always like thinking I shouldn't be doing. So even if you have to plan questions, do that. But yeah, once you sort of get into that habit of waffling, it's hard to get out of. And so just practice, practice, practice. For those like practice questions really help because it's technique, as opposed to like knowing content. I'm just trying to think about, the other stuff are just kind of general things. I'm just thinking about if I have any other study tips that I haven't mentioned. Again, visual things always worked really well for me. Hopefully I've answered most of your questions in the live chat, but they always worked for me, and I think that they helped consolidate information a lot. And sometimes I wouldn't understand things until I saw it visually. Again, you may be a different type of learner, but it might work for you. Flashcards, mind maps, all those sorts of things. Structure your notes around the study design. I don't know if I said that, but that's really, really important. Again, knowing the study design like the back of your hand, but structuring your notes around the study design can be really helpful because it can show you what is relevant and what's not. Creating mind maps linking the areas of study together as well, which kind of comes into that last dot point. Don't get too in the zone with one area of study. Like you don't be on immunity and think like, okay, I'm only talking about immunity. And then Vico trying to weasel in this little question about proteins to do with antibodies, but you're like immunity, and you don't realize that you're having to bring stuff in from other areas of study. I think I remember there was a question like that. I can't remember if it was on my exam, but it was about like photosynthesis and something to do with enzymes and then linking that to proteins. And I think Vico wrote like, a lot of students weren't able to make that correlation. So it's really important to not get too, like, to not like almost like segregate the areas of study. Okay, hopefully that all makes sense. Again, hopefully all your questions have been answered in the chat, but otherwise do feel free to email me. Again, lourdes@tutesmart.com. Hopefully that has all helped. Again, immunity is a big topic, but once you break it down, once you get through the processes, and once you just revise them constantly, I know this will sound like it's a little, but it just clicks. And I know it's easier like said than done just to wait for something to click. But if you keep on revisiting them, the process makes sense. And that's why I find like if you like I read about it, I drew it, I watched videos, I tried to get as much information from it as possible and kind of absorb the information in different ways so that it painted a bit of a bigger picture in my head. I think with stuff like this where it's very processy, that can kind of help. And yes, don't let experimental design fall to the back of your mind. Always keep that up to date. Same with the rest of Unit 3. Okay, I will not keep you any longer. I hope that all makes sense. Thank you guys so much for tuning into the lecture. Hopefully it's been a bit helpful, and good luck with all of your exams. Good luck with Bio 3-4, any other 3-4s that you're doing as well. I'm sure you'll do really great. Thanks, guys. Bye.